So good morning everyone, this is Dr. Shyma. Now, we come to the last class of vision. We'll be dealing with color vision today. The objective of today's class would be, will have to understand the physiology of color vision and also the physiology of color blindness. Color vision, if you look at, the human eye is sensitive to the light rays in the visible spectrum, and the visible spectrum basically has got 7 colors, and we call those 7 colors as spectral colors. And the spectral colors, or the 7 colors which we see in the visible spectrum, are violet, indigo, blue, green, yellow, orange and red, and they fall within the wavelength of 400 to 750 nanometers. Now, human eye cannot respond to a spectrum beyond this specific range. For example, ultraviolet rays falls, in the range of, or it's supposed to be greater than 400 nanometers. Sorry, lesser than 400 nanometers, and the infrared radiations falls under the spectrum of greater than 750 nanometers. Now, some of the animals are able to see the infrared rays, so their visible spectrum would be accordingly modulated. Now, the colors which are produced by mixing of 2 or more of the spectral colors, we call them as the extra spectral colors, for example purple. Now, human beings' eye has got the ability to identify more than 100 different colors. Color is simply a different wavelength of light reflected by object, and we have already told this. It falls in the visible spectrum of 400 to 750 nanometers. So you can see the visible spectrum here diagrammatically represented. You can see the same thing, the wavelength which of visible light which can be perceived by the human eye has been shown in a figure. The next slide talks about what color vision is. Now, color vision is basically the ability of the eye to discriminate between colors excited by light of different wavelength. And you all know that color vision is basically a function of the cones, and the sensation of color is very subjective. Now, when you perceive a color, or how do you see a color? Now, if you to perceive a color, or basically a color has got 3 different qualities. So basically the 3 qualities are intensity, hue and saturation. Now, in an abnormal color vision, what happens is one or more than one of these characteristics would have come down, either intensity, hue and saturation, which are considered as the 3 characteristics of the color. When they start showing variation, then it results in a defective color vision. Now, basically color, if you look at, I told you there are primary colors which are 3: red, green and blue. Now, any other color can be produced by mixing these 3 colors in different proportions. Now, these 3 primary colors, red, green and blue, when they are mixed in equal quantities, white color is formed. Now, what do you understand when the term complementary color is used? Each color will have a complementary color. When any color is mixed with its complementary color, the mixture appears white. For example, purple and green, orange and blue, yellow and dark blue are all complementary colors. So, now, when it comes to perceiving any color, another very important factor is played by the effective illumination, or how much light falls on that particular object, so that we perceive the exact or we perceive the color in its exact characteristics. So basically the color perceived depends not only on the wavelength but also other factors like illumination. Now when the spectral colors are seen in very dim light, the brightest part of the spectrum is always green, as the rods are more sensitive to green light. For example, in the daylight, the brightest part of the spectrum is always yellow, as the cones have maximum sensitivity to yellow region or the yellow light. So the brightest part of a spectrum shifts left during scotopic vision, and it shifts to the right during photopic vision. Now this shift, we call it as a Purkinje shift phenomenon. So that can be asked as a 3-marker. So here the slide also tells you, the 3 qualities, the hue, saturation or the lightness. Or lightness, what you mean is, how much of light falls on the object, or you can put it in more simpler words, the effects of illumination. Now to go to the next slide. Next slide talks about photoreceptors. Now photoreceptors, if you see the cones, cone shaped, they are less sensitive, they operate in very high lights, and the cones take care of color vision. Now the second important receptor of the eye, the rods, they are rod shaped, they are highly sensitive, they operate at night or basically dim vision, and they take care of the gray scale of vision. So the color vision is a function of the cones. You need to keep that in mind. So you can see a picture of the cones and the rods here. I think we have already discussed this part in the earlier classes about the photoreceptors present in the eye. This is a kind of briefing up where I take you all more into the concept of color vision. Now to go to the next slide, we just continue with it. Now the cones, you already know that they are more concentrated near the fovea, and it has the capacity to adapt to a wide range of illumination of colors and levels. When cones are concentrated more at the fovea, the rods, they spread throughout the retina, and any immediate response to illumination is a function of the rods, which is distributed all over the retina. Now the types of cones. Now the types of cones, if you look at, there are 3 types of cones: the L or the R, M or G, S or B. Now this L or R, that particular type of cones are more sensitive to red light, which comes in the visible spectrum of 570 nanometers. M or G, which is more sensitive to green light, which is in the visible spectrum of 530 nm. S or B, they are more sensitive to blue light, which is in the visible spectrum frequency of 430 nanometer. Now color blindness results when any of these cones are absent. Now if you stimulate all 3 types of cones equally, the result is white or no color, which I have already mentioned to you all. So you can see a graphical representation of that, which makes this concept more thorough for you all. So to go to the next slide, you need to know that there are different theories of color vision which exist. Now when you talk about the theories of color vision, there are 2 theories which exist. We broadly divide them into the trichromatic theory and the opponent processing theory of color vision. And this trichromatic theory is based on things happening in the retina, or it's basically a retina related theory, and the opponent processing theory of color vision is basically a neural theory. Now the trichromatic theory where, it's a theory which is also called as the Young-Helmholtz theory, and it was proposed by a scientist by the name Thomas Young, and later it was modified by another scientist, Helmholtz. So that's why you call it as the Young-Helmholtz theory. Now it's also called as, the retinal theory or the trichromatic theory, which I've already briefed up in the slide. Now according to this theory, or the theory considers as, there are 3 types of cones and each has a pigment with a different absorbance spectrum. Now the red sensitive pigment absorbs light maximally in the yellow portion of the spectrum. The green sensitive pigment absorbs maximally in the green portion, and the blue sensitive pigments absorb maximally in the blue violet portion. This I've already mentioned in the previous slide. This is, I'm just repeating it for a better understanding so that you relate to a theory and understand it. So basically the cones and the cone pigments are called red, green and blue respectively, and you call it as L, M and S. L stands for long wavelength, M stands for the middle wavelength, and S stands for the short wavelength. And the specific, the visible spectrum wavelength which they are sensitive to, I've already mentioned, the L, M and the S. Now for example, for this Young-Helmholtz theory, a green monochromatic light with a wavelength of 530 nanometers produces excitation of red, green and blue cones at a ratio of 31:67:36, and the person interprets it as green. I hope you get it. So I just repeat it. A green monochromatic light which is in the wavelength of 530 nanometers. It produces excitement of all the 3 cone systems, red, green and blue, but it's at a ratio of 31:67:36. Only then that particular color is interpreted by the person's visual cortex as green. Now at least 2 types of cones are required for color vision. Now according to the scientists Young and Helmholtz, color vision is due to the presence of 3 types of cones which maximally, or each maximally sensitive to 1 or 3 primary colors. So basically what you need to understand from here is a normal color vision depends on the integrity or the proper functioning of the cones. Absence or malfunction of one or more or one type of cone can lead to different types of color blindness. Now go to the second theory, which is the opponent processing theory of color vision, or the neural theory. Now this theory is also known as the Hering's theory of color vision, as it was proposed by the scientist Hering. He observed that there is no greenish red or bluish yellow color at all. The red and the green colors oppose each other, and the blue and the yellow colors oppose each other. Now the ganglion cells and the cells of the lateral geniculate body and the neurons of the visual cortex show the color opponent theory. This was stated by the scientist Hering. Now to go to the next slide. Now that shows the mechanism of the color vision. So just a second, I just would go back. Yeah. So now when we go to the mechanism of color vision, just have a look at the figure. I think I would just enlarge the figure a little. Yeah, you can see on the right hand side you can see the cones and the rods, and the next layer of cells you have the horizontal cells. Next you have the ganglion, large cells, and you can see the nerve fiber arising, which carries it, that's the retinal layer, and finally the impulse goes to the visual cortex. Now the P ganglion cells, they receive information regarding color from the cone cells, and they basically transmit it to the parvocellular layer of the lateral geniculate body. Now, impulse processing begins at the ganglion cell, where inputs from different types of cones are added, or input from one cone type is subtracted from the input from another cone. Now, the impulses which reach the lateral geniculate body are then transmitted to the deeper layer 4C of 6 and the blob region of the visual cortex through specific neural pathways. Now, what do you understand by the term the blob region of the visual cortex? Now, the blob region of the visual cortex contains clusters of cells that have very high concentration of the enzyme cytochrome oxidase. Now, if you look at a red-green pathway, it basically transmits the subtracted inputs of the L and the medium cone response. That's how it goes. If you look at the blue-yellow pathway, the difference between the S zone and the sum of L and M cone responses flow through it. And the luminance pathway that transmits the added inputs of the L and the M cone responses. Now, the information from the deep layer 4C of 6 and the blob of the visual cortex is transmitted to the V8 region of the visual cortex. I'll just repeat this concept once again in the coming up slide so that it's more clear for you all. Now, this whole thing what I've explained is, which this figure explains, is the mechanism of color vision. Let's just go through the points here. The medium wavelength causes the green receptor to fire. Now, as the wavelength gets longer and closer to 580 nanometers, the red begins to fire, surpassing the green. The short wavelength causes the blue receptors to fire. Now, therefore, color vision is a consequence of unequal stimulation of the three types of cones in a specific ratio, which I've already mentioned to you all. Now, let's go to the second slide, which talks about retinal processing. I think this I've already explained to you all, but we'll just go through the point. The color is mediated by ganglion cells that add or subtract input from one types of cone to the input from another type. So, that's seen in the figure. Now, going to the next picture, it's basically a diagram where the visual pathway is shown. I think there isn't much to explain here. Now, the pathway of color vision, if you look at all this, I have explained to you all, but I think we would just go through the points. I would just slightly enlarge this. Now, if you look at the pathway for color vision, the neurons carrying color vision in the optic nerve pass through the optic tract finally to reach the parvocellular part of the lateral geniculate body of the thalamus. Now, the lateral geniculate nucleus, or the it has got the different layers, the magnocellular layer and the parvocellular layer. Magnocellular layer 1 and 2 layers, the parvocellular layer 3 and 6 layer. Now, the layer 1, 4 and 6, they receive impulse from the contralateral portion, and the layer 2, 3 and 5, they receive impulses from the same side. Now, from the parvocellular laminas of the lateral geniculate body, fibers project to the blob regions in the layer 4 of the visual cortex. Now, I have already told you all what a blob is. They are basically clusters of cells which are arranged in a mosaic pattern in the visual cortex, and they take care of color vision. Now, going to the next slide about color blindness, what it exactly is. Now, the total loss of color vision is known as achromatopsia. Now, in partial loss of color vision, the inability to distinguish certain colors is termed color blindness, and generally 8% of men are affected and 0.4% of the women are affected by color blindness. Now, the most common type is red-green color blindness, and normally to describe color vision, there are different terms used, and you can see, I think I'll come to that. Yeah. So you can see, the classification. Oh, no, I just need to go back. Yeah. Now, we use prefixes like prot, deuter, and trite to describe red, blue, and green cone system defects. Now, this use of suffix. The suffix anomaly means color weakness, and the suffix anopia means color blindness. Now, if you look at this classification of color blindness, which is a very often asked 3-marker for you all, color blindness are classified into trichromats, dichromats, and monochromats. I'll just explain this in detail. Now, the trichromats, this is the most common type of color blindness, and about 6% of the affected males are normally trichromats. And basically, these trichromats have got all 3 cone systems present, but one of the cone system is defective. So, the ability of the individual to recognize one of the primary color would have really come down. Accordingly, the trichromats, have been divided further into deuteranomaly, protanomaly, and tritanomaly. This deuteranomaly is the most common one. Now, when we look at dichromats, 2% of the males affected by color blindness are dichromats, and basically the dichromats have got 2 cone systems and the third cone system is absent. And so the dichromat can be classified into deuteranopia, protanopia, and tritanopia, and again this deuteranopia is the most common one. Now, in individuals with only 1 cone system present, we call them as the monochromats. They basically cannot appreciate any color because minimum 2 cone systems are required to appreciate the color. So, a monochromat would see everything as black and white or shades of grey. So, that's been shown in this picture here. This is how the first pic shows. This is how the condition of dichromacy is. Second picture is a condition of a dichromacy. And the third picture is a condition of a monochromacy, where basically they see it only as pure black and white or shades of grey. So, the same picture is enlarged here. Now, if you look at, I think before I go to the test, yeah, fine, yeah. Now, the test for color vision, just one second. I think before I go to this, I would like to tell you all a little more on this. Yeah. Now, basically, the color vision, color blindness, that they have been categorized into a trichromat, a dichromat, and a monochromat. And basically, what you should understand is this color blindness is it can be either inherited or acquired, or even due to acquired by any lesions or due to any drugs. Now, the color blindness is inherited as an X-linked recessive disorder. Now, as the males contain X and Y chromosomes in their germ cells, they develop color blindness if the X chromosome contains a defective gene. On the other hand, the females develop color blindness only when both the X chromosomes are defective, which is normally very, very rare. The females having one defective X chromosome remains symptom-free, but they can transmit the disease to half of her sons. Thus, the females are carriers and the males are the sufferers. Now, all the daughters of a colorblind person becomes carriers and half of their sons suffer from the disease. So, basically, color blindness manifests in every second generation males. Deuteranomaly and protanomaly are the most common defect. Tritanomaly and tritanopia are rare and occur both in females and males. Now, sometimes, following a lesion of the striate cortex, which is area V8, the individual can suffer from complete loss of color or what was it? The complete loss of color vision called as achromatopsia. Certain drugs like sildenafil inhibits the enzyme phosphodiesterase, and that can also cause color blindness. So, let us go to the next slide, which talks about the test for color vision. Now, the test for color vision is divided into 2 categories. the pseudo isochromatic chart test, where we have the Ishihara's plates and the colored dots, which will comes under that category. Then you have the Hallman-Wool test, which has a color matching from selection of skeins of colored wools. And then you have the Edridge green lantern test, and the last one is spectroscopic test. Now if you look at what, get to know what the Ishihara's chart is, yeah, you can see, the Ishihara's plate there. So basically the Ishihara's chart, it's a most routinely used test for color blindness in the clinics. And the Ishihara charts, it consists of lithographic plates in which numbers or sometimes pathways are drawn in colored spot against a background of spots of different colors which are of different sizes. Now the subject reads the number and also traces the pathway. The colors of the numerals or tracings and the background colors are arranged in such a way that a colorblind person reads them differently or traces a different pathway when compared to what is done by a normal person. I think I'll just go back to the previous slide. Yeah. So that's about the Ishihara plate. Now you can see, the Edridge green lantern test. In this test, the light from a lantern is shown through different colored glass piece fixed on a rotation disc, and the subject is asked to name the colors. Then you have the Holmgren's wool matching test. The subject is asked to perform a series of color matching from a collection of wools of different colors. Now color blindness is usually done as part of a health checkup for entering into professions like driving, flying, traffic services, railways, armed forces, etc. And when there is loss of visual acuity, color vision is also affected. But basically in conditions of bilateral lesions of the inferomedial occipital region or the optic neuritis, color vision, you can see, gets more affected. So this is how it is. Let's go to the next slide. This is one of the Ishihara plates. So can you see a normal person would read it as 7-1 with all the yellow and brown color dots of different sizes, and this is how he perceives it, a normal vision. But a colorblind person, which is the condition of achromatopsia, you can see that he cannot see any color in the Ishihara plate, and this is how he normally sees the world around him. So this is basically the Ishihara chart. I think this would make things more clear for you all. Can you see the first picture where it's number 6 with all the different colors of green, yellow, brown and orange? That's how a normal person sees it. A protanopic would see the same thing as in the second part. A deuteranopic would see it like this, and a tritanopic sees it like this. This is how the Ishihara plate would be read by people with different conditions of color vision. Now you can see color blindness. A normal person would see it like this. A red green blind would see the same picture as it's shown in the second one. A blue yellow blind person would see the same that normal thing in this color distribution. Now the importance of color vision, I have already told you all. It's required for different entering into different professions like government jobs, driving, flying, traffic services, railways, armed forces, even medicine. So that's it. I think we wind up with color vision. I think so. There we come to the end of vision. Thanks for listening. Please make sure that color vision is very thorough for you all. It's a very often asked 5 marker or a 3 marker, or sometimes you can include this even as a part of your 10-mark essay when visual pathway is asked. So thank you guys. Thanks for listening.